Imagine finally being told, that there might be a functional cure for a lifelong chronic illness.
Right, which is huge.
It's incredible. But, and here's the catch, to prove it actually works, your doctors have to take away every single medication currently keeping you stable.
Yeah, and then they basically just watch you walk a tightrope without a net for 6 months.
Exactly. It's wild. Welcome to the deep dive, everybody. Today is June 26th, 2026, and we are looking at a massive phase 3 trial that asked patients to do exactly that.
We've got our hands on a brand new, highly anticipated article straight from the New England Journal of Medicine.
And the stakes, I mean, they really could not be higher. We are looking at the trial results for a new treatment. It's called Bepirovirsen.
Yeah, Bepirovirsen.
And it's targeting chronic hepatitis B virus infection. But the authors aren't just looking for better symptom management here.
No, not at all.
They are explicitly investigating the potential for a functional cure, which is, the holy grail. So, okay, let's unpack this because the medical terminology here is pretty dense.
It is, yeah. Very dense.
And to really appreciate the sheer audacity of this trial, we need to understand the actual weapon they're testing.
Right. So the paper defines Bepirovirsen as an antisense oligonucleotide targeting hepatitis B virus transcripts.
Okay, which sounds incredibly futuristic.
It does. But it fundamentally comes down to intercepting the virus's internal communications. Because when hepatitis B establishes a chronic infection in the liver, it acts like, kind of like a rogue factory.
A rogue factory.
Yeah. It embeds a master copy of its genetic material right into your cells and continuously creates these transcripts.
Like little chemical blueprints.
Exactly, blueprints. And those blueprints tell your cellular machinery to just churn out more viral particles.
Okay, so standard treatments kind of tackle this from the back end, right?
They do, yeah.
Because the patients in this study were already taking stable nucleoside or nucleotide analog therapy.
Which we usually just call NA therapy, to keep it simple.
Right, NA therapy. And from what I understand, NA therapy works by basically feeding the virus faulty building blocks. So the viral factory tries to build new copies of its DNA, but because it's using these defective materials provided by the drug, the whole assembly line jams up. The virus can't replicate.
It jams the assembly line, but, and this is the key, it never shuts down the factory itself.
Ah, okay.
The blueprints are still being printed. That master copy is still just sitting there inside the liver cell, constantly trying to restart production.
So the moment you stop taking the meds?
The moment a patient stops their daily NA therapy, the factory just grabs healthy building blocks again and goes right back to work.
Wow.
This is exactly why standard treatments are a lifelong commitment.
Which brings us to the new drug, the antisense oligonucleotide.
Right.
So if NA therapy is sabotaging the building materials, is an antisense drug basically like intercepting the factory's mail and shredding the blueprints before they can be read?
That is a perfect way to look at it. It is a synthetic strand of genetic material designed to hunt down and bind to those specific viral transcripts.
Shredding the mail?
Exactly. Once it binds to them, the cell's own enzymes recognize this, like unnatural pairing, and they destroy the transcript. The blueprints are shredded.
Okay, and shredding those blueprints achieves something NA therapy really struggles to do, right?
Yeah, it cuts off the production of viral proteins.
Yeah.
Most notably, the hepatitis B surface antigen, or HBsAg.
Let's talk about that surface antigen for a second, because losing it is a strict requirement for what this study considers a functional cure.
It's the ultimate goal.
Right. The trial defines a functional cure as having unquantifiable viral DNA in the blood combined with the complete loss of this surface antigen.
Sustained for at least 24 weeks.
After a fixed duration therapy, which means after all the therapy has ended. Why is shedding this specific protein so critical?
Well, because the surface antigen isn't just a structural part of the virus, it's basically a smokescreen.
A smokescreen?
Yeah. The virus pumps out these massive, overwhelming quantities of this antigen into the bloodstream, way more than it actually needs to build new viral particles.
So it's deliberate.
Highly deliberate. The purpose of this excess protein is to flood the body's defenses. It constantly triggers the immune system until the specific T cells designed to fight hepatitis B basically become exhausted.
Oh, wow. They get overwhelmed by the sheer volume of targets and just shut down.
Exactly.
Yeah.
They fall into a state of deep immune exhaustion. The virus uses this smokescreen to hide in plain sight.
That is insidious.
It really is. So by shredding the blueprints with Bepirovirsen, you stop the production of that surface antigen. You clear the smoke.
And the hope is that by clearing the smoke, the patient's own exhausted immune system can wake up.
Wake up, recover, and finally clear the remaining infection on its own.
Man. And that leads us to the Be Well trials, where they actually put this theory to the test. Because taking a patient off daily medication and relying on their immune system to suddenly take the wheel, that requires a bulletproof trial design.
Absolutely bulletproof.
So they set up two replicate double-blind trials, Be Well 1 and Be Well 2. And the demographic profile was highly specific. We're talking adults with non-cirrhotic chronic HBV.
Meaning their livers hadn't progressed to severe irreversible scarring yet.
Right. But they had an established active chronic infection. This was indicated by a baseline surface antigen level of more than 100 to 3,000 international units per milliliter.
And importantly, every single one of these patients was actively taking that standard daily NA therapy.
Right. So what was the actual regimen they tested?
So they used a 2-to-1 ratio. The experimental group got an injection of Bepirovirsen at a dose of 300 milligrams every week.
Subcutaneous, right. Just under the skin?
Yeah, subcutaneous injections for 24 weeks.
Yeah.
A straightforward fixed duration treatment.
Okay, but, and I have to push back on the timeline here, if they only took the drug for 24 weeks, why did the study require eligible patients to completely discontinue their stable NA therapy at 48 weeks?
Yep.
And then wait all the way until week 72 to measure the primary outcome?
What's fascinating here is the sheer confidence and the risk of that design.
It's terrifying.
It is. By stopping the baseline NA therapy at week 48 and waiting another 24 weeks until week 72, the researchers intentionally remove all pharmacological support.
Taking away the safety net.
Exactly. They are demanding proof of the sustained part of the functional cure. If the antisense drug didn't truly wake up the immune system to maintain control, that 6-month window with zero medication gives the virus ample time to come rushing back.
It's like building a bridge, taking away all the scaffolding, and then, parking a fleet of heavy trucks on it for 6 months just to prove it won't collapse.
That's exactly what it is.
Taking a patient off their stabilizing meds is such a massive gamble. So did the bridge hold? Or did the virus come rushing back?
Well, looking at the primary outcome at week 72, the contrast is stark.
Night and day. Let's look at the Be Well 1 numbers. Out of 650 patients who received the actual drug, 127 achieved that functional cure at week 72.
Right.
That is 20%. And in the placebo group, out of 328 people, 0.
Absolutely 0. And the replica trial, Be Well 2, mirrored those results almost perfectly.
Oh, wow, really?
Yeah, out of 570 patients on Bepirovirsen, 106 achieved the functional cure.
So 19%.
19%. And in the placebo group of 286 patients, we again see 0 functional cures.
Okay, I have to play devil's advocate here for the listener.
Go for it.
A 20% success rate is fascinating, but we need to address the glaring math. To a layman, 20% might sound like an F on a test.
Sure.
80% of the people taking this intense cutting-edge drug did not achieve a functional cure. 4 out of 5 patients went through weekly injections, came off their standard meds, and still saw the virus rebound.
Yeah, they did.
So why is this being hailed as a significant field-altering victory?
Because you have to anchor your expectations to the baseline reality of chronic hepatitis B.
Which is 0.
Exactly. The fact that literally 0 patients in the placebo groups achieved a functional cure proves conclusively that spontaneous recovery for this demographic simply does not happen.
Period.
Period. Standard NA therapy does not cure this disease, it only manages it. A jump from an absolute 0% chance of a cure to a 19 or 20% chance of a cure is a paradigm shift. We have moved from impossible to 1 in 5.
Okay, that makes sense. But what is happening in those 4 out of 5 patients? If the drug shreds the blueprints and clears the smokescreen, why doesn't the immune system wake up and finish the job for the other 80%?
This gets to the most insidious survival mechanism of the hepatitis B virus. We mentioned the master copy of the virus sitting in the liver cell.
Right, the factory.
Well, in many patients, especially those who have had the disease for decades, the virus takes its genetic code and physically integrates it into the host's own DNA.
Oh, man. So it weaves itself into the patient's own genome.
Yes. The human cell itself becomes hardwired to produce viral transcripts.
So it's not even a separate factory anymore.
No, it's part of you. Bepirovirsen is excellent at clearing out the circulating transcripts. But if the viral DNA is heavily integrated into the host genome, the moment the drug leaves the system, the host's own cellular machinery just prints thousands of new blueprints.
The factory is too deeply embedded.
Exactly. The 20% who were cured likely represent a population where the virus hadn't integrated as deeply, allowing the awakened immune system to actually win the biological battle.
The biological battle. And that perfectly sets up the catch here, the cost of this success, because the source material is remarkably transparent about the safety profile, and the journey to that 20% cure rate is pretty brutal.
Not an easy road.
Not at all. Looking at the pooled analysis at 72 weeks, 91% of the patients in the Bepirovirsen groups reported adverse events.
Yeah, 91%.
Compared to 73% in the placebo groups. And serious adverse events hit 7% for the drug group versus 4% for the placebo.
And the clinical reactions during the actual treatment period were substantial. When we look at adverse events of grade 3 or higher, which are severe medically significant events, they occurred in 16% of the patients receiving Bepirovirsen.
That is almost 1 in 6 people.
Yep, compared to just 3% on the placebo.
This is where the physiological mechanism becomes crucial to understand, because the text specifically highlights that the most common grade 3 adverse event with the drug was an increase in the alanine aminotransferase level.
Right, ALT.
ALT. That happened in 6% of the patients on the drug. But ANT is a liver enzyme. It's a fundamental marker doctors use to assess liver damage.
It is.
So if the whole point of this trial is to save the liver from hepatitis B, why is the drug causing severe spikes in the exact enzyme that signals liver damage? How do we square that circle?
I'm glad you caught that, because it seems entirely counterproductive until you realize that the ALT spike isn't simply a toxic reaction to a chemical.
It's not.
No, it is the sound of the immune system going to war. When liver cells are damaged or burst open, they spill ALT into the bloodstream.
Okay, let's rework the way we usually think about this. Let me try an analogy. Imagine you have a severe termite infestation inside the walls of your house.
Okay, I'm with you.
The house is your liver. The termites, the virus, have pumped out a chemical pheromone that puts the local exterminator to sleep. That pheromone is the surface antigen smokescreen we talked about earlier.
Spot on. And then Bepirovirsen comes in, shreds the blueprints, and suddenly the pheromone is gone. The smokescreen lifts.
Right. And the exterminator, your own immune system, wakes up.
Yes.
It looks around, realizes the house is completely infested with termites, and it goes into a frenzy. It grabs a sledgehammer and starts smashing holes in the drywall to kill the bugs hidden inside.
The drywall is your infected liver cells.
Exactly. And the debris pouring out of those smashed walls, that's the ALT enzyme spilling into your blood.
That is exactly the biological reality. The drug doesn't just quietly sweep the virus away. It triggers a massive, localized immune response.
The immune T cells are finally recognizing and attacking the infected liver cells.
Right. So the spike in ALT is severe, and it requires careful medical management, but it is often a necessary sign that the immune system is actively clearing the chronic infection. It's collateral damage from a waking immune system.
Wow. You don't get the miracle without the mess.
You really don't.
It forces a complete reframing of how we view adverse events in these kinds of trials. A severe side effect isn't always a sign the drug is failing. Sometimes it's the chaotic evidence that the drug is doing exactly what it was designed to do.
Which is why understanding the mechanics of a trial like Be-Well is incredibly valuable for you, the listener.
Oh, absolutely.
Even if you don't have chronic HBV, it transforms how you consume medical news. When you grasp the mechanics of antisense technology, the insidious nature of viral integration, and the biological warfare causing those ALT spikes, you look past the sensationalized headlines.
You see the bigger picture.
You understand the profound human cost and the complex biology required to move the needle from a lifelong daily pill to a true, sustained cure.
Moving the needle from 0% to 20%. I mean, we started this deep dive looking at a rigorous, high-stakes trial that intentionally stripped away the safety nets of standard medication just to prove a cure could hold.
Yeah.
We explored how intercepting genetic blueprints can lift a viral smokescreen, and we weighed the reality of an awakened aggressive immune system causing severe liver stress in the pursuit of healing.
It's an incredible balancing act.
It really is.
Yeah.
And it leaves me with a thought I just haven't been able to shake since we started parsing this data.
With that?
Well, if a 20% success rate, accompanied by a 16% rate of severe adverse events, is the new gold standard for a massive field-altering breakthrough in chronic viral treatments, it forces us to ask, has pop culture entirely ruined our expectations of medicine?
Well, that's a great question.
Are we so conditioned by movies and television to expect a glowing 100% magic bullet cure where the patient takes a single pill and wakes up perfectly healthy without a single side effect, that we fail to appreciate the incredible, messy, and incremental miracles that are actually happening in science right now.
It's definitely something to think about.
Something for you all to mull over. Thank you for joining us on this deep dive, and we'll catch you next time.